Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha, Senior Life Science Journalist at Xtalks.com. And this week, I am happy to introduce a special guest on the show, as always. This week, we have with us today Dr. Adam Grant, who is a principal scientist at CancerVax's, a company developing a novel immunotherapy-based cancer treatment system called the Universal Cancer Treatment Platform. The system uses AI to train the immune system to detect, mark, and kill cancer cells. And unlike other immunotherapies, it actually modifies cancer cells. And we'll learn more about this new innovation. Very exciting. And I'm personally very excited to learn more about it. And so Dr. Grant is one of the inventors of the Universal Cancer Treatment Platform. He's an expert computational biologist specializing in the use of next-generation sequencing data to identify new drug targets and biomarkers of response to cancer therapeutics. He previously served as a bioinformatician at Xenter Medical Technologies and as a computational scientist at RAPT Therapeutics. His innovations include software designed to improve cancer antigen detection, as well as a gene signature approach for selecting treatments for breast cancer. He earned a Bachelor of Science in Bioinformatics from Brigham Young University, as well as a PhD in cancer biology with an emphasis in bioinformatics from the University of Arizona. To learn more about innovative immunotherapy approaches to treating cancer, including, of course, the work that Dr. Grant is helping lead at CancerVax's with its innovative new platform, let's turn to him now. Thank you so much for being on the show.
Thanks for having me, Ayesha. Excited to talk to you about the work we're doing at CancerVax.
Likewise. And so I'd like to learn more about your journey and what drew you to the company, to CancerVax, and if we could learn a bit more about the company's mission and goals before we get into all of the other good stuff.
Yeah, I first was introduced at CancerVax about 6 months ago, and the idea of what they were trying to do really resonated with me. And looking back, it is a simple idea, but it's just not something that anyone's really tried to do, and that's just because of this mRNA technology. Now we can talk about that later, but really, what we're trying to do at CancerVax is we're trying to trick the body's own immune system to kill the cancer cells, by making them look like something that the body can easily recognize already, such as like measles.
That's awesome. And so really, I guess having learned about this novel idea, but like you said, it's simplistic in theory, but to actually apply it and see, oh, wow, like why didn't, we think of this or something like that. So I can imagine how you were drawn to the company.
Yeah, exactly. And it's just like you said, how do you do it is the
Mm-hmm.
really the difficult part. And what we're doing a lot of development in right now and testing. And so, and a lot of it is, I mean, it is unfortunate that it happened, the COVID-19 pandemic, but a lot of the research that we are diving into is because of what's happened since then with these nanoparticles, LNP delivery of it, this mRNA technology. And it's just really, in my opinion, I think it's a game changer for the cancer therapy field. And it's still being so explored. It's so early on right now, but it's just so exciting to be at the cutting edge of using that mRNA technology to treat cancer patients.
Indeed. And so, could you tell us a bit more about the Universal Cancer Treatment Platform? And I also love the name because it's simple, to the point, and it's letting people know what it's out to do.
Yeah, and I'll be honest with you, we are a very ambitious group at CancerVax, and we believe that our technology could benefit practically all cancer patients in the future. Of course, we're still very young, long ways to go, but we're very hopeful, and that's because it's just the idea, you know. if we can make cancer cells look like something the body can already recognize, it should be able to work on every cancer type. And of course, there will be some caveats that we need to overcome, but that's the idea. Is it's going to be a universal cancer treatment because we're trying to make every cancer cell look like, you know, measles or something. But it comes back to the delivery. And so we have a unique approach where we refer to as like a marker one and a marker two strategy. Our marker one strategy focuses on proteins that are expressed on the outside of a cancer cell. And so our marker one approach will go and attach, and this will vary across different cancer types. So there's not going to be one specific treatment that we can apply to every single cancer. There will be these variations of this marker one of what surface protein we target on the outside of the cancer cell, whether it's breast cancer, pancreatic cancer, ovarian cancer. And then also our marker two strategy, which further distinguishes between healthy cells and cancer cells to make sure we're only targeting or much more biased towards cancer cells.
Right. And that's an RNA based approach where you go in with.
Yeah,
Sorry,
yeah,
go.
exactly. Sorry, I get really excited here when you start throwing out words like that. So yeah, and what we call it is our smart mRNA technology. And so the idea is that our marker one strategy will help our therapy get into the cancer cells. However, there's just not a single protein that can completely discriminate between healthy cells and cancer cells. And that's a big problem. And chemotherapy is the best example. I mean, it's just unfortunate that no matter what, you're going to get a lot of off target effects, and that causes a lot of secondary symptoms and adverse side effects. And we're really trying to eliminate with this, marker one, first trying to narrow it down even further to the cancer cells and potentially some other healthy tissues that express the surface protein. And then using our smart mRNA technology that distinguishes between cancer cells and healthy cells based off of their genetic signatures inside
Inside.
of the cell.
So it's like a dual pronged approach where
Yeah.
you're identifying the cancer cell based on what's on the outside and also what's on the inside.
Yeah, exactly. Perfectly said.
And then the other step is then to mark these cancer cells. So you want to make them look like you said, like measles, so that the immune system can recognize them. And I think this is such a brilliant approach in terms of, or something that people have not really focused on, because the focus has been on the immune cells, right? How do we modify the immune cells to
Yes.
in traditional immunotherapy approaches? It's all about taking a patient's immune cells, modifying them, making them more powerful, injecting them back in and unleashing them against the cancer. But then you still have the same problem where, well, are they going to be, how do they really effectively identify and target the cancer cells? But here you are modifying the cancer cells. Tell me more about that.
Exactly. What we like to say here at CancerVax is, it's very difficult, near impossible to teach a dog, an old dog, new tricks. And that's kind of what every other immunotherapy is doing, is they're trying to teach the immune system what cancer looks like. And I mean, there has been some great successes in the
Absolutely. Yeah.
immunotherapy field, but there's still this problem that it's just not effective in every single patient. There's still it, it's very lacking in that regard. And so that's why we're coming from this different approach. Is the immune system is already really good at killing things that already knows. It just can't recognize cancer cells, unfortunately. So that's where we're going to make the cancer cells look like something that the immune system already knows.
And how do you achieve that by injecting using lipid nanoparticles? Are you tagging, sending in an mRNA, and that will express a measles specific protein?
Yeah, exactly. And then it marks, and then the immune system can detect it, and then it will do what it's been doing for billions of years, is to eradicate those viruses.
That is, again, such a neat approach. And I was so excited to learn about it when I was researching the company ahead of this interview and the technology. So it's so cool. And as I think, you touched on, that there are current gaps in conventional or current immunotherapy approaches in cancer treatment. And so, how does the platform, as we talked about, is taking a different approach, but what are some of the preliminary data or readouts that are confirming that this is an approach that can be effective and can truly be universal?
Yeah, and for us, like we've already have done so much development, and we have done some research on our end already, and showing one of the main things that we shared with the community a couple months ago is that we were able to get this smart mRNA working. But in terms of looking at the efficacy, or how can we stimulate these immune cells, that's something that we're currently working on right now. And the idea is that is other immunotherapies, they only work in certain environments. And so what we like to say in the immunotherapy field is that the tumor needs to be hot already. There has to be immune cells in the tumor microenvironment for these immunotherapies to work, uh, much more often than not. And that's why you see it like in melanoma is one of the best responders of immunotherapy, because melanoma, they have tons of mutations in them already, and there's these immune cells gathered there. So one hypothesis that we have at CancerVax is that I think we're going to be able to benefit patients that have a cold tumor much more readily than those immunotherapies. It's just because those cold tumors, they just don't have those tons of mutations that say, "Hey, I'm a cancer cell, come and kill me," such as some other um, immunotherapies do. Like checkpoint blockade is what anti PD-L1, anti PD-1 inhibitors. But for us, it by it's kind of like a security system. If we can have set the alarm off in these cancer cells, by making it look like measles. I the can't the immune system's not going to know that it's a cancer cell. It's just it knows it's a measles infected cell, and it's going to have to get rid of it. And so by doing that, I think we could really take advantage of these cold tumor microenvironments, allowing us not only to potentially help patients that have a hot tumor microenvironment, but go into a new patient population that immunotherapies just don't work in. Yeah. And of course, these are hypotheses. These are what we're testing, but based of current literature and our understanding, I mean, if we feel like it's going to work. So
That's so great and truly very exciting. And so, in terms of the cancers, you did mention some cancers and indications that that may benefit from this, but yeah, actually universal, because the idea is that whether it's a hot tumor, whether it's a cold tumor, and also would it equally work in solid tumors versus liquid tumors like leukemias, although um, I've worked in AML, and so my supervisor always taught us that AML is a solid tumor of the bone marrow. So there's contention there whether it's a liquid tumor or a blood cancer or not, but there's that.
So, and right now we're focusing more on solid tumors, just and mainly because there's a much more clinically unmet need in a lot of these tumors. And and of course, like you said, universal cancer treatment is what we hope for in the future, but you got to start somewhere. And so you know, where we're starting right now is pancreatic cancer. Is we feel that these patients could benefit the most. And I mean, I'll be the first one to admit pancreatic cancer, it's a very hard to treat cancer. There's already been so many other therapies that have failed in this cancer type. But we believe, because of this unique situation of pancreatic cancer, where it's kind of hot. It's not cold, it's not hot, it's kind of warm. And so it has these immune cells, but they do not respond to current immunotherapies such as checkpoint inhibitors. And interestingly, there's been a paper that came out a year or two ago about these. personalized cancer vaccines, which has
Right.
been really popular. And they're a bit different from us because what they're doing is, it's similar to what we talked about before. Is they're teaching the immune system what a tumor looks like, because what they do is they get a biopsy from the tumor, and then they sequence the mutations in that tumor, and they make a personalized vaccine for that tumor. And so, and what was shown in this study is that patients benefited quite, I mean, for pancreatic cancer, it was great. 50% of, patients did respond to this personalized cancer vaccine. However, there's a lot of problems with personalized cancer vaccines. One, they're very, very expensive. They just not might be accessible to everyone. Two, they can take a long time to make.
Right.
I mean,
Yes.
like 8 weeks. And I mean, when you have pancreatic cancer, you're
Yes.
already fighting against the clock. And the biggest one that I get the most scared about is there's not really any good biomarkers for these personalized cancer vaccines. You responded, why? Who knows? It's just, after, would you pay like so much money for a therapy with 40% success, 30, maybe 50? I don't know. There still needs to be a lot of research done, but there's just no guarantee. And then, of course, it extends your life, but it's still unsure of it, like, how much of a benefit for that cost. But at CancerVax, we're trying to overcome those problems by more of this universal. And some people might say, "Oh, universal. That I would much rather get more of a personalized approach." But if it's universal, I mean, that just means it can work for everyone at a much. It's much more feasible.
Yeah, I think there's been obviously a huge focus and emphasis on personalized medicine, especially in oncology, with the advent of, obviously molecular technologies, DNA, RNA sequencing, and all of that. Um, but I think taking a step back and looking at the bigger picture is often necessary. And like you said, it's often not feasible to go after each type of tumor. Each person's tumor is different, essentially, in many cases. And so I love this idea where it's like a plug and play system. We were talking about this during COVID
Yes.
about the vaccines, right? The mRNA vaccines. And that's kind of the beauty of it, where you can plug and play different mRNAs, different antigens here. And time is of the essence, and really being able to adapt this technology. And in a way, like you said, the antigens will be different for different cancers, obviously, but in terms of the targets that they're going to identify. But at the end, if you're taking them with just something universal like measles, then yeah.
Exactly. About 95% of the population has received a measles vaccine. And I mean,
I was going to
we're
ask you
thinking
about,
about
yeah,
chicken pox
yeah, or
too. Yeah.
chickenpox, yeah,
It doesn't just have to be measles, thinking of some other approaches
hmm
that we can think about.
or perhaps
But
even.
that's what we're doing. Is we're sorry, Ayesha, but we're targeting, these viruses, or not targeting. We're trying to make the cancers look like viruses that are so prevalent in the populations, to help with that universality of our approach.
And it just makes sense to do so. Um, I mean, there are concerns about measles outbreaks happening now because of, I don't know if we want to go there, but lowered vaccination rates and decreased uptake, just given the climate and environment that we are in, because post COVID and even before, with vaccines and hesitancy around them. So, how do you look at that angle, or is that something maybe down the road, or is that a consideration that is being talked about at CancerVax? Or
I mean, we're talking about it, we're thinking about it, but and everyone has their own choice. Everyone can make
absolutely,
their
yeah, we're
decisions.
all for choice. We're not saying, yeah, we're not shaming
Exactly,
anybody, or
but
yeah.
and that's something we're just more, going as, looking at the environment. But you know, regardless, measles is it's just a really good contender because, first, even if you don't get yeah, maybe we shouldn't go quite there, but it's just for most of, and I will admit there might be some very unique situations that people, you know, we might have to work around. Like maybe if someone like for our therapy, there's even an option of, and this is something we're still debating. It's not, it's just something we're considering is that maybe even we could give like a booster shot before the patient receives our therapy. And that's something that's really nice you can do with measles. And then to say, if it might, in order for our therapy to be more
To yeah,
effective, this is something you might have to do.
have to do right. That
But
makes
once
sense.
again, these are
Those
still
are
we're still
considerations, yeah,
these
down
are
the
just
road,
things
but
we're
yeah,
thinking about, yeah,
for sure, yeah.
and more about. So
And in terms of really, so when I was in grad school, bioinformatics was a field that was really up and coming. And now I can't even imagine where it is now with, AI and all of these other machine learning tools and all of that. So, as a bioinformatician in cancer biology, how have you seen the field evolve and where is it now and where do you think it's going?
yeah, it's been amazing. So and it for me, every day I work with data, and so I am very limited to the type of data that is accessible. And so I can only do so much with the data that's available. The good news for people in computational biology right now is the amount of data we're getting is becoming exponential. And the community is just it's really great in the cancer field because a lot of people publish um the, these papers and they share this next generation sequencing data with us. And maybe like a lot of times these authors they're using this data for something, but then I'm able to come in and tweak it a little bit and utilize it for a question that we have, even though it wasn't the focus of the initial paper. And there's just so much more of that data coming out. And what's really nice for us is that we're a small company. We can't afford to make a lot of costly big mistakes. But at CancerVax, because with my computational biology background, is we're able to narrow down so many variables that we might want to test, and especially in designing, like what is going to be the best marker one that we should use for a specific cancer type, or how should we design our smart mRNA to really prevent off-target effects and healthy tissues. And this data is just it's not something that was there 5-10 years ago, but it is now, and that we can utilize that to quickly design these things. And I will admit it's just amazing how fast we've been able to move, and we are already you know creating smart mRNAs, testing all of these different components, and we're really hoping that, by early next year that we could start hopefully seeing some of our results in some mouse models.
That sounds amazing, really. Again, to be able to extract insights from data that previously you may not have been able to without the technologies now available to us. It's opened so many possibilities, and it's just yeah, amazing.
Yeah, exactly. And it's just, and I feel really blessed. And with the team at CancerVax, I'm the only computational biologist, but it's so nice, and we have some great biologists there. And so for us to be able to communicate, where I say, "Hey, I see something in the data. This looks really interesting. Have you? What are your thoughts about that?" Their biology is much stronger than mine, but with us working together, being able to parse all of this data and use these machine learning algorithms, and it's a really cool environment to be in.
I can imagine. And I know that as a computational biologist, you were also working in looking at gene signatures. And is that something that's also involved in the approach, or is it you're just looking at, let's say, the top ranking gene expressed in a certain type of cancer? Or could it also be, would it be one marker, or could it be more than one based on signatures?
Exactly.
Yeah.
How do you combine markers? And that's the question. How many markers do we need to use? And depending on their marker one or marker two strategy, it varies. And it might vary for different cancer type. But because of all this development we've already done at CancerVax, we can easily identify those, answer those questions really quickly. And so we could hurry and design something that we has a much higher probability of working rather than us just going through the literature and trying to see what other people did. And of course, it's more often than not their situation is much more different than what you're or the context that they're trying these different variables. But just being able to unbiasedly look at the data like that, I think it's really giving us a leg up.
Absolutely. And actually, I'm just thinking about this, even though it's a universal platform, however, you still are looking at, patients, individual, tumors, and you will be going in and seeing what the optimal targets are in terms of designing that smart RNA and target
Exactly.
to, right? And so, and this is combining both, universality and personalization at the same time, actually, in a way.
Exactly. Yeah, there and it's I
These are also just
it's
terms I think now that have, kind of get overused, maybe, but
yeah, but you know what? But for me, I'm in a really unique situation because I don't think a lot of people think about it this way. But like, I'm I probably get to know patients better than
I
maybe
just
not as good
wanted
as
to
a
ask, yeah,
doctor.
that was going to be one of my next questions in terms of, I ask all of my guests about patient centricity because that's so important, the patient perspective. So, how do you incorporate the patient, in your work and be guided by that?
It's it's very difficult, and one thing that I've been able to see probably more than a lot of other scientists, just because I just look at the data so much more, is the heterogeneity that exists in these patients. And it's just so hard. Each patient's tumor is probably as unique as they are, and and it's just all the this data that I'm using came from someone that it's just like such a blessing to me that this patient was willing to give their sample so that we can use it for the future. And it's like, and seeing just how unique, and it is hard. A lot of my computational career has been trying to find similarities, and there's honestly in cancer, there's not a whole ton, not as much as you would expect, just because of this variability, and that's a big problem in the therapeutic approach. It's like, and that's why chemotherapy is still the first line defense. It's just because it just works. It hits everything. It's just unbiased in that regard. And so, by taking this approach with at CancerVax of having this, you know, going more of tricking the immune system, I think we can, still incorporate these unique patient data, but take advantage of a common trend that a lot of us have, which is a functioning immune system that already has these memory CD8 T cells just sitting there patrolling the body. And no other immunotherapy is trying to activate these memory CD8 T cells. They're just trying to create these new, naive CD8 T cells. Exactly. Yes, exactly.
Here you're leveraging what the body already has, and it may not be faulty, right? Because I think the sort of mindset or perspective oftentimes in immunotherapy is, oh, maybe the immune system isn't functioning optimally, but that's not really the case. It's like they're trying to boost what you already have, but maybe what you already have can be leveraged in a different way by, yeah, in terms of making the cancer more available and seen.
Exactly. And that's what, and cancer is just so good. I mean, at hiding, escaping. It has all of these tools to do that. And that's why it's just such a lethal disease.
And do you see this something like this? Do you see this platform being beneficial for recurrence and that as well? Because you'll be able to interrogate, like you were saying, the heterogeneity is immense in cancer, and so are you going to be looking at different subpopulations of cells and which ones may be responsible for recurrence or for metastasis or for it gets so complicated. It gets complicated real fast, I
It
know.
does. But this is what excites me about the immunotherapy field the most, is because if for cancer is able to hide from the immune system. However, once it starts to kill the tumor, they can have something called a systemic response. It can start the immune system with like, hey, now I know what a cancer cell looks like because I've already after it's some of them have already died. And this is one of why immunotherapy can be so amazing is because once the cancer cells are revealed to the immunotherapy, it starts killing them and it says, hey, I'm learning what a cancer cell is. And now I can start killing them, maybe even if they don't have, they look like measles,
Right, right,
you
right.
know.
Oh, okay.
And then it's like, there's a metastasis over here. And I know those are cancer cells now, so I'm going to start killing those. And so that's why you get these amazing complete responses in immunotherapy. And we're hoping that at CancerVax that our approach is just able to really, you know, stimulate that effect by just really, I like to think of our approach as like a really good defibrillator. Just give a huge shock to the tumor and it's like, hey, wake up, immune system. Like, we're gonna help you. We're gonna give you this shock, and then you you should be able to take care of the rest. I mean, hopefully. But of course, I mean, how many treatments will we need? And how that those are things we're still answering. But that's how I see our approach.
Yeah, no, absolutely. That's again, yeah, it's such a great way to put it. Of course, you're training the immune system, so it will be able to recognize um, the cancer cell even without the measles component. So
Yeah,
yeah,
exactly.
that's the
Mhm.
beauty of immunotherapy there. Absolutely.
And so, but just recently, we have just really escalated
Hmm. No, no,
in
that's
terms of our research, our goals, and we meet together all the time. We're always talking. We're always sharing ideas with each other. I mean, we're all so passionate about what we do at CancerVax. I think our wives were maybe a little less passionate about what we do, but we're working and just sending emails on the weekend. But we're just so excited about the
the
possibility
possibility.
of our approach.
Absolutely.
It's really good.
And I can only imagine that there's definitely interest within the industry. And when we talk about partnerships and collaborations, can you tell us about some of any collaborations or partnerships that CancerVax currently has or may be looking at in the future?
Yeah, exactly. And so, and so what we're trying to do, I mean, we don't have all the capabilities, we're a virtual company. And so we depend on people to help run our experiments and and we're not a lipid nanoparticle company. We don't have the capabilities to make an LNP. And you know what? The best part in today's world is we don't need to. There's already so many great companies like and so right now we're working with Cytiva. They used to be formerly known as Precision Nanosystems. And anyways, but they have a great lipid nanoparticle formulation, and we are working with them to use that in conjunction with our smart mRNA, and then to work with them to adding in our marker one approach on the outside of their lipid nanoparticle so that it can, more specifically go to these cancer cells. And then, we're working, and so I'm in the US, Utah, Cytiva's in Canada. We have
We're in
we've teamed
Canada
up with
too. Sorry.
awesome. And then we have, we're working with Access Biosciences, and they're in Northern Ireland, and they're running all of our experiments. And we actually did quite a bit of research. We talked to a lot of vendors. That was just like for 2 months straight. It's like we have this great idea, but is the infrastructure even there for us? And then it's like, okay, well, we found Cytiva, we found Axis, and one of our most important vendors is TriLink. They're making this synthesizing our mRNA for us. And so we're able to come up with these ideas and to, really, I mean, we do as much in terms of reading papers. We read everything we can about this topic. We're always analyzing data and coming up with these ideas, and then we're working with these collaborators, which it would just be unfeasible even if we were a huge company to be able to synthesize our own mRNA or to create our own LNP. And and the lab space is just so expensive. But by collaborating with these individuals, I was just blown away when we were first doing this. It's like, wow, the infrastructure is really there for us to move quickly and to really help patients. Like, and a lot of these, ideas, they're already validated to some extent. We're just putting it all together in such a unique way. And so that gives us a lot of confidence in our approach because we're always, you know, we are the most cynical at CancerVax. We're always thinking of like, what if this happens? What if that happens? How can we overcome that? And as a scientist, it seems like you went to grad school. You have to be, and that's just, and because we've all been there. So we're doing everything in our power to overcome that.
Yeah, you
So
just want to make sure you're covering all all
yes,
possibilities, and within that,
exactly.
the possibilities can go awry, and yeah, especially
We
coming from
are our biggest critics at CancerVax.
exactly, exactly. And that's the beauty of science. It's always evolving, through failure or through, through both failure and successes. So,
Yep.
yeah,
So
it's
we're trying to eliminate the failures as much as
yes, we try
possible.
to eliminate the failures as much as possible,
Yeah,
but
no promises though. But if there's always a learning experience when it
exactly. And you
happens. So
learn the most from, those setbacks, and yeah, you only grow from
yeah,
that. So,
unfortunately, I wish I knew everything already, but I'll admit I'm not there yet. So
no, well, maybe you have until 5 p.m. today, no? Be a running joke in the lab, right? It's like, okay, I'll
yeah,
have
exactly.
we'll have the cure by 5 p.m. Friday.
It's funny. When I was in graduate school, I went through a daily cycle of waking up in the morning, having an amazing idea, thinking I'm the smartest person in the whole
Right.
world.
And then you
Go
look
to
it
lab,
up.
testing it.
Yeah, or something, yeah.
Yeah, go home and realizing I'm not the dumbest, but maybe top. But anyways, and then just repeating that cycle over and over again.
Yeah,
So
or that someone's already thought of it, you start, going up on
yeah, that's
PubMed and
yeah.
you're like, "Oh, okay."
Man, I'm not as cool as I thought I
Yeah.
was. Being the
It's like, "Damn it, I
person
was
to
scooped."
yeah.
From my brain to
5 years
it just
earlier.
yeah. They just got to it earlier.
Yeah.
But oh my
So
gosh.
it was,
But yeah, and in terms of I think we've talked a lot about, of course, the fields of cancer immunotherapy, how far it's come, and how far it looks like it's going to go with the kind of work that you're helping lead at CancerVax. And so, where do you think the field is headed? Again, with all of these approaches, computational biology, AI, the biology being better understood, novel approaches. So, what are kind of, I hate to say trendy, but what are kind of the things that you think are going to lead the evolution and immunotherapy revolution forward?
revolution. Yeah, good way to put it. I'm a big believer of this mRNA technology, and of course with having AI helping, but really mRNA technology I think is going to lead because it just opens up so many different avenues that we could have just never imagined. And it's the way that I think about it is that the body is already so good at what it does. A lot of times, it's just this unfortunate event. You have a mutation of some extent and you get cancer. But the body, we've been around, it's evolutionary. And mRNA allows you to really, you know, utilize or tweak or take advantage of these
Mm-hmm.
molecular pathways that are
Yep.
just a disarray. Unlike any, and you can, there's just it's so hard with small molecule, um, you know, other forms. Antibody drug conjugates, just antibodies, they're, and of course, I'm not saying that mRNA doesn't have its own hiccups, the biggest one being delivery right now. And that's something that we're really actively working on at CancerVax. But just, you know, there's nothing better than just being able to correct something that's wrong with the body with its own tools. mRNA, it's just oh, there's so it's just in every cell, so many copies, billions of mRNA just floating in your cell. And it's just I didn't like we mentioned this before, but with the COVID-19 um pandemic, it's just really unfortunate, but it really just opened up the way for this therapy. And I don't know, like, my background's in cancer, and so I see a lot of mRNA technology that can be helped in the cancer field. But what about other diseases? Like, I can only imagine that, because every disease is caused by some sort of
Genetic
dysregulate.
underlying, yeah, factor, yeah.
Yeah, exactly. And of course, and like, yeah, CRISPR I think can help a lot too. But even it's just the ethical, safety. But mRNA is just it's nice 'cause it's just temporary. Like you don't have to worry about really these long lasting effects. It's either it kind of worked or it didn't. And how do you make it more potent? That's things that can be overcome with mRNA technology.
Yeah, I think absolutely. It was so eye-opening and so exciting to see like this molecule that, we learned about, worked with in grad school, and then all of a sudden during the pandemic everyone's talking about it. It's like
Okay.
everyone's like saying mRNA. I was like, "Oh wow, this is like so crazy and it's so cool." And like you said, it's opened up so many possibilities. And we've gone a step up from the DNA level at a
Yeah.
more at a level that's more, I think, it has more utility because it's often obviously
Yes.
the complications that come with modifying DNA, and like you said,
Yeah. Exactly.
the ethics and all of that. But if you but so a step up from that, you have this ready to go molecule that can be tweaked, modified, introduced, and you're on your way.
And
And
it can
it's
be
a level step
synthesized.
down from the protein level, which is often a little bit more also complicated to or complex
Uh
to
huh.
deal with, target
Yeah.
in terms of, so
And it's just so accessible now
accessible. Yeah.
too.
Yeah.
Virtual company, we can go to TriLink and say, hey, we want this sequence. And they're like, great, we'll give it to you in like 6 weeks. I was like, this is so amazing. Like, it's just so wild. This is the really the possibilities. We're really probably the ones that are holding ourselves back with what it's capable now.
Hmm. I love that. Well, I'm not love that, but it definitely makes
So,
sense. Yeah. And in terms of milestones from CancerVax, I know, it sounds like there's so much going on. So what can we expect in the coming months and years? And what are some key goalposts or milestones that you're looking to achieve in the near future?
yeah, we have a lot in the works right now. And it's really interesting with our approach because, as mentioned, we have a marker one and a marker two strategy. But what's really exciting about this is when we were trying to, develop a plan of getting these, we realized that we could easily make these in parallel. And so simultaneously, we are, optimizing our marker one approach and our marker two. We're designing our smart mRNAs. We're testing. We've already validated that smart mRNAs work. We're just optimizing it for pancreatic cancer right now, and then further, tweaking it if we need to. And then at the same time, we, working with Cytiva to making sure we can get that LNP, the nanoparticle, and can we
Mm-hmm.
it keeps encapsulate mRNA, not necessarily our smart mRNA right now, but that will go in later. And then also, can we at, add our targeting moiety onto our LNP? And so we're hoping within, the next 3 months is that we'll have updates on, are we able to specifically deliver a nanoparticle to tumor cells that express a specific surface protein, and, prevent or making sure that healthy cells that don't express that surface protein don't get that, you know, our payload. And then also, is our smart mRNA going to work inside of pancreatic cancers? And it is able to really distinguish between healthy tissues and pancreatic cancer. And then we're going to combine them together and then start, making sure, are we able to look at the biodistribution in mice, making sure is this nanoparticle
Right.
going where we want to and only expressing like a luciferase protein, just something that glows, we just want to make sure that it's really preventing off target effects. It's critical for us. And then further we'll incorporate our payload. Can we really make these cancer cells look like measles? And also making sure that they're only targeting the cancer cells and not any of the other healthy cells, or much less biased. And so really we're hoping that by early next year that we'll be able to start actually testing the efficacy. You know, are we able to, I mean, you got to see it in mice. Are we able to get rid of these tumors in mice? Do we see a reduction in size? Are we, helping these mice live longer, at least, that have gotten tumors?
Mhm. And
And then, of course,
just so maybe if our listeners didn't catch it, but this would be an injection that you would give um, to,
yeah, we're,
yeah,
we're hoping we will be testing different administration routes, but we're hoping it's going to be an intravenous
intravenous.
injection. Yeah,
Okay.
so it'll just be, mhm.
Okay. As opposed to like a sub Q or whatnot.
Yeah,
But
exactly. Mhm.
okay.
So, and that's the idea, hopefully it'll just flow throughout your body. And
Mhm.
we're hoping that this gives a lot of benefits to not only the primary tumor, but also
Yeah,
the metastases
that's key.
that are there.
Absolutely. Well, Adam, it was such a pleasure speaking with you today. And thank you so much for your time, for your insights, and for sharing this really exciting technology that you're helping develop at CancerVax. I am personally so excited about it. And, I was talking to some colleagues here at Xtalks as well. And they're like, oh, wow, that's such a cool approach. And so I think really you have something special here. And I wish you and your team all the best, and we'll definitely be keeping our eye out for all of those milestones, and we'll love to have you back to share them.
Yeah, I had a great time, Ayesha, and you just asked all the right questions. It was so great to talk to you, and I really appreciate everyone that's listening to this. And yeah, we'll be keeping you updated. We try to keep our community involved as much as possible. I think I'm more on the science side, but I know that you can sign up for our newsletter at CancerVax to
Great.
keep
Yeah,
updated. And of course, we would love to, continue talking to X Talks
absolutely.
anytime. It was
That's great to know. Yeah, so check out CancerVax's website where you can sign up for their newsletter. I know I will be doing so. So that's great. And yeah, we'll definitely be in touch. And I wish you all the best. And thank you once again.
You too, Ayesha.
All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.